Hi everyone, welcome to Foresight's Biotech and Health Extension seminar series sponsored by 100+. We're delighted to see so many of you here. I just want to put one note in before we hit the road in terms of housekeeping. We have Vision Weekend coming up, which is our annual member gathering at the end of this year. And it's basically like a festival, one in France in a castle outside of Paris, and then one in San Francisco. And it's basically bringing together the entirety of the community, not only our longevity track, but also our neurotechnology, space track, our intelligent cooperation track, our existential hope track, basically lots of other technical tracks that we have at Foresight. And we're collaborating with fellows, funders, sponsors, and people that just want to join the community anew. Basically, we'll discuss all of the different technological breakthroughs over the year, how we can advance them, what are potential risks, how we can address them, and so forth. And so if you're interested in not only having like some longevity exposure, like in these seminars, but if you're interested in meeting many people in person, either in the castle in France or in San Francisco across 3 different areas, then please do join us. There's a very strong longevity cohort always and a big longevity track. And I'd be delighted to see many of you there. And I know that many of you are already coming, but I just want to put it on your radar that tickets are going relatively fast. So if you do want to come, now is a great time to get one. Okay, that has been it from me. Now I want to hand it over to Shahaf, and I'm so delighted to have you here. We have quite a lot of people in the waiting room again. So I have the sense that this could be quite a talk that generates a lot of questions. So for everyone in the audience, if you want people to be able to upvote your questions, if you want to make sure that it gets asked, then drop it in the chat as Shahaf is presenting, because then people can upvote it. Otherwise, you can also raise your hand after the discussion's over. For now, I'm very excited to have you here, Shahaf, and you're going to discuss Turn Your Mito On. So very excited to have you here, and I'll share more info about you in the chat, and we'll discuss a little bit more about your context within Foresight afterwards, I'm sure. But for now, the stage is yours. Thank you so much for joining, and I'm excited for the Q&A.
Thanks, Alison, and thank you very much, and for Foresight for giving us the opportunity to give this talk. Yeah, I cannot really compete with the location of Castle in Paris, but I'll try to do my best with this picture. So I know a couple of the people that already are part of the list of the listeners, but a lot of people I don't know. So I'm looking forward to also have discussion and get to know new people. But there are 3 things I know about everyone that is listening to this talk. So first of all, each one of you recently ate some foods, whether it's today or some extreme people yesterday. The second thing, all of you now are consuming oxygen as you're listening to my talk. You do it constantly, even without noticing it. And the reason you do it is you burn the food you just acquired to make ATP, to make energy to live. Without it, you will die. And the third thing is that actually it's not an ability, it's something we cannot do. None of us here in the audience can utilize an energy that is basically all around us. And of course, I'm talking about light. And this is what I'm going to talk about today. It's something we, Luminova, try to change. And it's using a new technology, human technology, to bring together 2 conceptual ideas of metabolism that have been separated by 1.5 billion years of evolution. Let me start by saying, who are we? So first of all, he's also in this conversation, Andrew Wojtowicz. He is one of the co-founders of the company, but importantly, he is the one that actually discovered or invented rather the tool that or the technology that we're going to discuss today. And I know Andrew is already eager to answer all your questions at the end. Then there is me. I'm also co-founder of the company Luminova Biotech, and I'm interested in aging. I come later to this technology when I was trying to look for something funky to do in aging. And we have Peter, who is our seed investor. He invested in us in this crazy idea to have exclusive licensing on our own patent, ironically enough, and to enable us to start doing some operations with the company. Luminova sounds actually very Russian, I just realized, but what is really the company is about? So what is our mission? I'm going to talk about it later, just going to give some pointers at the beginning. We want to treat aging by harnessing the energy of light into our mitochondria. And this is a rather disruptive technology. As far as I know, when I go to conferences and meetings, nobody talks in this direction. And we want to use this disruptive technology as an energy replacement in aging. And I'm going to talk about it later. As a proxy, and that's going to be at the end of my talk or the last part of my talk, we want to translate what we found in lower organism into a mammalian system, and we will talk about the eye and eye diseases such as diabetes at the end. I also want to say what is not the mission of Luminova is not to use some intervention that have already been studied a lot, diet intervention and exercise or inhibitors for proteins. which within us, or like activating some pathways which are within us. We want to do something different. And why do we need to go a bit disruptive or crazy and out of the box? And this is probably Neil Barzilai is now laughing. He heard me saying it already a couple of times. This is the perspective by Jan and Brandon, and I really like it. And it's a bit pessimistic to everyone in the aging field. And the idea, they say, there is a limit to maximum lifespan, and interventions, such as the ones I've mentioned, diet, exercise, and so on, they probably don't have any evidence for maximum lifespan intervention. That's what they claim. And they say, if you really want to break a glass ceiling, so if you really want people to live more than 120 years, we probably need to do something radical to fundamentally change the aspect of our human biology. And somehow it echoes something that I believe in. And so what can be disruptive? What can be something out of the box to tackle aging? So I went to a conference a while ago, and I asked the students, give me some ideas how you can extend lifespan. And almost everyone surprisingly said something related to lifestyle. So be it, don't smoke, don't use drugs, don't drink, sleep. But then I forced the question in a sense, give me something crazy, something that has not been tested, at least at that time. I think it was 2018 or so. And then more crazy ideas came into the game. And some people say insert genes from long-lived animals. I think actually Vera recently showed it from Rochester University. Some strange drug cocktail, drinking young blood. I think Brian Johnson is going in this direction, not drinking, but transfusing from his kid. Freezing, like cryopreservation. Some people, I think, saw a lot of the Terminator movies, so they said machine-human interface. And my idea was actually more modest, and it was eating ATP, our cellular coin in the mitochondria in our cells. And the reason was, initially I thought in a lazy term that it's basically being lazy. I felt like we invest a lot in making ATP and we generate some sort of problems when we do it. So I just thought, wouldn't it be wonderful just to have the end product? So just to eat the ATP. And here my supervisor back then, when I told him that, he said, Shahaf, forget it. You need to eat 70 kilos of it. It will not cross the membrane. It's charged. It's not stable, so it will break down. But I'm not the person that gives us so easily. So how do we do it? So you cannot really do it, but maybe there is a way around it. So this is an illustration of a person, and you can really appreciate, even if you look at yourself in the mirror, how much of your body is designed to eat food, to seek food. If you don't have it, you start to be agitated and angry, and you look for it. And a lot of organs in your body are dedicated to handle the foods, deliver it, process it, and so on. And at the end, when these metabolites, or what's generated for the food in terms in forms of metabolites such as glucose, we all heard about it in biochemistry classes. So when you have the glucose inside the cell, it's having a lot of different reactions that generate acetyl-CoA, and then it goes into the TCA cycle. And all this process lead to the end, to the electron transport chain. And I'm going to zoom in on that because it's the heart of the talk. So if you look at the electron transport chain, you have the four complexes. And what they do is they use the metabolites that are generated by the TCA cycle to generate this proton gradient, or the PMF. And it's against the gradient. So the cells know in the mitochondria to move protons across the membrane against the gradient. And by that, it generates some sort of a force. And this force can be used that the protons then find their way back into the other side of the mitochondria membrane via ATP synthase, and every 2 to 7 protons move the ATP synthase enough to make ATP. And this is absolutely essential, as I mentioned. If you see a James Bond movie with cyanide here that block the oxygen consumption, then you die almost instantaneously. You suffocate. And why was the rationale to target or think about this something which is so conserved, not in human, but basically all eukaryotes? The rationale is, first of all, a lot of people already show that targeting mitochondria can lead to modulation of lifespan. Also, aging itself is accompanied by mitochondrial dysfunction. But I want to talk a bit about different angle, and this is a very basic concept. So if you have nutrients that you eat plus oxygen, to burn that in the mitochondria, you generate the ATP energy. And Andrew always like to say, this is very much like, you can think about like a factory that makes electricity. If you take carbon and you burn it with oxygen, yes, you generate energy. The problem is that on the way, you make a lot of pollution or a lot of damage. And the same, I think, is applicable in our cells. When we do this, we have a lot of products that we don't want to use, and it accumulates in aging. So this is one angle to look at that. So what can you do about it? So this is here the electron transport chain, use oxygen and move protons, and you have ATP synthase. And here I really looked for something to work on that. I actually walked in the same direction when I came across Twitter on Andrew's paper. So what Andrew and his PhD student at the time, Brandon Berry, they invented a tool that is called mitoon. And the mitoon, I'm going to talk in the next slide. What it can do is it can harness the energy of light to do the same principal idea that the electron transport chain. So a little bit about mitoon, it's a game changer in my mind. When I saw this paper by Andrew, I really fell in love with this technology. So this is a transmembrane protein that is engineered in the mitochondria. When light hits it, it drives protons. It does it through a cofactor called all-trans retinol that is also produced in our body. And I think what's really interesting that the mitoon is derived from fungi. So that's also something to consider for future translational studies. It's not a protein that exists in us or in any mammals for the sake of the story here. It's actually come from a metabolic concept that is, as I mentioned, 1.5 billion years ago separated from us. And what's Andrew's brilliant work was to take this protein that pump protons in fungi, but not in the mitochondria. He engineered it in a way so it goes inside the inner membrane of the mitochondria in the right orientation. And what it does now, it's generate this proton gradient with light. And this is Andrew's work in EMBO reports in 2020. So just to a small piece of data here. So he showed that if you isolate mitochondria, you can generate ATP with the mitoon just in the presence of light without any substrates. And whereas if you take the isolated mitochondria with substrates, you can generate the same amount of ATP, but with less oxygen. And this is where I come into the picture. I really thought this was amazing and it has to be tested in aging. We tested it in aging. So we tested it on worms and we tested different conditions with and without light. Worms do not have ATR. So this is the cofactor for mitoon, as I mentioned. So you need to add it to the food. And first of all, Brandon was the first author. He showed that this mitoon insertion by itself is not toxic. It's very important because sometimes when you express proteins in the mitochondria, it is toxic. And this is straight to the kill. So we try to see if it extends lifespan. And indeed, we show that if you have ATR plus light in the mitoon, you have a substantial lifespan increase. Now, it's not only the increase of maximal lifespan and median lifespan, but also what it does, if you look at worms at the same age, at the age of 4 and the age of 10, not only they live longer, but they are also physically more active. And I think this is very important because some lifespan intervention may have trade-offs in terms of fitness. So it seems our worms are long-lived and they are more active. We also show that whereas light is enough, a certain amount of light, if you use too little light, there is basically no lifespan extension. So there is a lot of room still to understand what is the minimal requirements of light, what is the maximal that is not toxic, and so on. And this, I think, is just a proof of principle that if you use FCCP, that dissipates, it's uncoupling, it's an uncoupler for this PMF. Then if you use this, you cannot generate as much as proton gradients. You don't have this lifespan increase. So it's specifically this pathway. And lastly, because I think mitoon is a new player in the field, we tried also how it will react alongside another way to extend lifespan. For instance, AMP kinase. So we know AMP kinase activation of it increased lifespan. And we see that both of them together results in a very interesting and large lifespan expectancy. And if you want to read more about the paper, I really like the cover of this Nature Aging spotlight that they've done. You can read more in Nature Aging we published early this year. To summarize this part of the talk, so I think I don't want to go too much into the theory behind it. If someone asks, I can explain what is this biphasic theory of aging. But I think it's also the mitoon might have different benefits in different time points in life. So in early phases of aging, I think in early phases of aging, what's happening is we try to generate always the same ATP, but it's starting to get harder and harder. And we invest more metabolites and more glycolysis, for instance, to generate the same amount of ATP, maybe even consuming more oxygen. But by doing so, we generate more cellular damage. And the mitoon in this aspect can partially replace this need to increase. So we'll achieve the same amount of ATP, but using less upstream reactions, including oxygen consumption. Whereas in late phases of aging, where our ATP is decreased because mitochondria is very dysfunctional, their mitoon can just compensate for the reduction and bring it back to more useful like ATP levels. And this has been accepted for publication in Trends in Biochemical Sciences. Yeah, you can read about it. I just want to also talk about some of the limitations of this technology. So it is going to be gene therapy. And we need to understand how to deliver it to have a critical number of cells. Some tissues are more easy than the others, but we need to hit enough cells to make enough impact. This is one of the other hurdles, is how to have light accessibility. The eye is very easy, and we're going to talk about it in a second, but what about the brain and the liver and so on? For sure, we will need to make toxicity assessment. We're going to introduce a protein that has been far away in evolution. And it's going to be likely a chronic treatment. So it's not going to be a magic pill you use once and that's it. It's going to be likely chronic because we will need to have constant assistance of mito to our metabolism. So yeah. So the goal of Luminova is not really to increase lifespan of worms or flies and such. That's not really what we have to. We want to translate it into mammalian models. This is also Andrew's work, Zukim from Rochester University. And what they have shown is that the concept of mito also works in mammalian cells, for instance, HeLa cells in human and CD8 T cells in mice. And you can see that if you use mito specifically with light, you have an increase of ATP both in mouse and a human model. So we are very encouraged by that, and we want now to move forward. So one thing, and this is something I'm still educating myself, we want to look at mammalian eye and aging. So I guess it's quite obvious if you look in the literature that during aging, there is more and more prevalence of getting some vision problem in the eye. It happens already a bit at the age of 65, but you can see when you hit 75 and onwards, a lot of us will have problem with the eye, and that's very important organ. It makes our life much easier if we can see what we are doing. And what we want to look at mito, if it can be a preventive approach, treatment, or a post-treatment. And the reason is why the eye is, well, first of all, as I mentioned, eye and light is basically what the eye needs to do. So delivering light to the eye is very easy, and it's not going to really change so much our lifestyle, it's just around us. But I noticed when I did the literature search, there are a lot of problems that can occur with aging in the eye, diseases, AMD, cataract, and so on. And what I saw that not all of them, but many of them has some connection that can be benefited by mito treatment. So mitochondrial dysfunction is something that has been described in the aging of the eye quite often. Cataract, very famous problem with vision loss, or distortion is one of the things that contribute to cataract is oxidative stress. And also the eye is one of the organs that you really see a lot of accumulation of lipofuscin. It's a byproduct that is accumulates and we cannot get rid of it. And it's a problem for our cells to handle this. And it's also somehow to some extent generated by oxidative stress and reactive oxidative species. So the idea is what I just mentioned, if you use mito, you can first of all tackle the mitochondrial dysfunction, you can generate more ATP. You also probably, this is something we need to test in the mammalian system, you probably will lower the oxidative stress because we are less dependent, as I mentioned, on oxygen and upstream metabolic reactions. And hopefully this is something we really want to test now. It will reduce the accumulation of lipofuscin, giving the likely to be less reactive oxidative species as we use less oxygen and upstream reactions. And another thing we want to do really in parallel in mice is diabetic retinopathy. And this is something that probably a lot of us know. A lot of people have diabetes. But what's really interesting that if you look around the world, the people that have vision problems due to diabetes is actually quite substantial percentage, accumulating to millions of people that suffer some sort of extent vision loss in diabetes. And I think here mito will be superb to try to tackle this disease. Because of the following. So first of all, again, we see that mitochondrial dysfunction and oxidative stress alongside accumulation of toxic waste as AGEs is contributing factors to this diabetic retinopathy. And what's really also interesting is when you have diabetes, you have excess of glucose that goes to small capillaries in our eye, blocking it and killing these capillaries. And that produces the state of hypoxia. So that means we do not have enough oxygen in our eye to generate ATP, which is a major problem. However, the great thing about mito, it can do the same trick even when there is less amount of oxygen. So we think here, it's something we have to test now. We will try to add miton to diabetic model of a mice, whether by AAV or we generate a transgenic mice, and we try to target mitochondrial dysfunction, hypoxia, and AGE production. So this is something we have set to start. We already now contacted several CROs, precisely 3 companies. We're going to talk to them in the next 2 weeks. By preliminary talks, it seems like the funding for this type of experiments will cost us around $300,000 to $400,000. We really want to make sure we have enough controls and we have enough base proof of principle to move to clinical trials. I also had the opportunity to talk to a company that does clinical trials. Of course, this is a bit more down the road, but this is, of course, going to be more expensive step. And the other thing is, I guess I don't see the questions yet, but I guess some of you ask, okay, great. Maybe you can treat the eye, maybe the skin, but he mentioned before brain and liver. What's the idea to actually target that? So we have the skin and it basically doesn't let the light go inside. So we have a couple of ideas. I cannot talk too much about it here in general. If someone is really interested, they can come to me and talk to me later one-on-one. We try to explain more. There are 2 main ideas. The first idea would be to shift the light sensitivity. Miton or the upgrade of miton can be more sensitive to light spectrum, which is more reddish, and it can penetrate a bit more. I'm a bit skeptical how much it's going to work. The other thing is we try to, I think this is more crazy idea and promising idea. What we want is to generate light inside our body in a druggable system and to generate the light close enough to miton so it can use the energy of light to move the protons. But this is something which is still really early stages. We started to work on that. I hope in the next 2 years, we can talk about it a bit more in detail. So just in summary, I think at the end, we want to use this upgraded technology to show it can extend also lifespan through targeting inner organs in our body. And this is something I think which is really the future beyond the eye and the skin. So with this, I thank you very much for your attention. I didn't want to do 40 minutes talk because I think in 28, 25 minutes, you can get everything. I want to open it now for discussion, and Andrew will join me. Thank you very much, and looking forward for discussion.
Wonderful. Thank you so much. That was really interesting. First question from Cosmo. You go.
Yeah, we could probably take it offline. I've got too many questions regarding beta carotene processing in the mitochondria, and I'm wondering what your thoughts about that are. Because there are enzymes in the mitochondria in humans that do process beta carotene into retinol. And there's some weird connections between those enzymes and the Steve Horvath clock that I've been trying to figure out.
Yeah, Andrew, so you want to go ahead?
Oh, you didn't hear me?
No, I got it. So, yeah. So I'm Andrew. I'm
hi.
Collaborator with Shahaf that helped design this technology. So, yeah, you're correct. The metabolism and turning beta carotene into the retinoids, which are going to be important for a lot of metabolic enzymes. So the initial studies that we did were in C. elegans, in which that didn't play a role at all in that lifespan extension. And that's mostly because C. elegans, they don't have a photosystem, they don't have eyes, they can't see light. So that's why we needed to supplement it. So in theory, even if that metabolic processing of beta carotene into retinol acid is damaged, we can supplement that in addition to overcome those deficiencies.
Okay, thank you.
And again, I'm happy to follow up offline too if you have more specific questions about that pathway.
Yeah.
Okay, John, you're next.
So the next one up there says we propose to develop. Oh, sorry, go ahead.
Yeah, you proposed to develop a drug that would create light inside the body to drive the mito on. But where does the energy come from to create that light? Wouldn't you have to burn more fuel to power those drugs that are creating light?
So excellent question. So if we think about systems that we already know that generate light, so think of a firefly and luciferase, that comes with a price. And you're right, that consumes ATP. However, there are newer systems which use drugs, and the energy is found in that drug. So that drug is broken down by an enzyme. It will just give away light without consuming ATP. So there are systems out there that will allow us to generate light just by adding a drug, which essentially will have no energetic cost.
Okay, so as I understand it, you'd have to keep taking this drug almost continuously to provide continuous light.
That's correct.
Can you quantify energy output by grams of drug?
Can we quantify how much energy is coming out? So I guess we would have to do some of the testing, and we can actually see it. So it's used a lot in microscopy. So where people will use these systems to generate light and subcellular compartments. So for example, this technology has already been used to illuminate mitos within a cell. So we would be able to quantify the light from that system.
Thank you.
All
And
right.
then somebody also posted on there too, there are other systems that we can do that are by doing implantable LEDs or again, longer wavelengths of light, which can reach deeper inside the tissue.
And a related question.
Yeah, go for it.
I understand that when you shine light between 750 and 800 nanometer, you have a good response in the mitochondria and the eye already without any additional changes or drugs. So can you comment on this topic?
Yeah, so that's actually a really interesting field. So they use very long wavelengths of light, so almost infrared light. And what they'll do is they'll shine light on systems, and there's no known biological target right now for that system. We don't know why that happens. Now, the current hypothesis is that it's targeting mitochondria in one shape or another. However, there's very little, again, mechanism behind it, and we don't know much about it. In our system, we have a biological target. We have something that's druggable that we can, again, eventually create a system which directly activates one particular component. Again, the long wavelength light, the infrared light system, is thought to heat up the system, and is supposed to make mitochondria, they think, work faster, which in some scenarios may not be a good situation if your mitos are already dysfunctional.
Okay, next one up we have Aram. Do you want to ask the question yourself, or do you want me to ask it? And Alexandru, you got another response from Cosmo in the chat.
And ask the question. So my question is the orientation of the protein is crucial in that aspect. If you have a reverse orientation, you actually will do a huge damage, because then there will be no ATP production whatsoever. So my question is there a chance that could happen? Now, there is also another side to that. The mitochondria, it has a tendency for fusion and these type of activities. There is a potential also for creating some disorientation or whatever. I don't know if you actually theoretically thought about this possibility.
So excellent question. So right now we have the orientation, so it will mimic the electron transport chain called mito-on. The technology that our company has patented, we've also created the reverse. So we call it mitochondria off or mito off. So that one is flipped around the opposite direction through different targeting domains as well as structural domains to lock it in one position or the other. And so as you allude to, some situations it may be good to supplement mitochondrial function. In other scenarios, it might be better to just completely shut them down. So as you alluded to, mitochondria are dynamic. They undergo fission and fusion. And what the idea really is that if we can target this to damaged mitos to help them achieve that proton motor force to make ATP, we can selectively increase those damaged ones and help them. And that's some of the technology that Shahaf was talking about, that next version of mito-on. We can use sequences that direct proteins specifically to damaged mitochondria to help supplement them so that they can go about making more energy. And that's something that we're looking into the future.
Yeah, and you're relying on the expression in the nucleus or in the mitochondria. I didn't get that.
So the way mitochondria have their own DNA, plasmid DNA, as well as there's mitochondrial encoded proteins in the nucleus. So mitochondrial DNA is heteroplasmic. There's lots of copies of it. Where this is encoded is in the nucleus. And so, it's nuclear encoded, it gets expressed, and then there's a leader sequence, which again, I'd say about 99% of mitochondrial proteins are going to be expressed in the nucleus. There's very few proteins that are encoded in mitoDNA. So when it's encoded in the nucleus, there's a leader sequence which pulls it to the mitochondria, and then it's imported in just like all the other proteins.
Okay, thank you very much.
You're welcome.
Next one up, we have Creon. And Micah, if your question is still relevant, feel free to... you answered it in your head. Good. Cool. Creon, you go.
Thanks a lot. Very interesting presentation. Does this stuff, either in the initial light harvesting form or the targeting that you just mentioned for mitochondrial regeneration or repair, does it relate to mitochondrial uncoupling in some real way? And if so, can you discuss that?
So that's exactly like the previous question. If we flip the pump the opposite way to the construct that we call mitochondria off, that will uncouple the oxidative reactions from the phosphorylation reaction. So if we had mito off, it would turn off mitochondrial function. Mitochondria, on the other hand, will mimic the reactions of the electron transport chain. And so it should decrease our reliance on consuming carbon and on consuming oxygen. So it's not necessarily uncoupling in the canonical bioenergetic system. It's decreasing mitochondria on is decreasing our reliance on consuming oxygen.
Yeah. Okay, that's understood. But as I understand it, what goes by the name mitochondrial uncoupling in the metabolic literature is not turning mitochondria off. It's causing them to free run more like they do in, say, brown fat.
Yes, exactly. That's like an uncoupler such as FCCP or dinitrophenol DMP. Those are drugs that will uncouple the redox reactions from the phosphorylation reactions. Mito off will mimic that, but mito on will not. Does that make sense?
Some sense, yeah, but we can leave it here. Thanks.
Okay, and just to go on with those uncouplers, like those are drugs. There's no way to turn them off. They freely diffuse everywhere. Again, like for DMP, that was the first drug that was the FDA was invented to ban that drug. But this is a controllable mechanism to generate energy.
We have 2 more questions. I don't know, Carl, if you want to unmute or if you want me to ask the question.
So how much can you crank the system up under ideal conditions, right? So you're replacing some fraction of the normal energy production with light. What percentage can you replace? And they're like ideal in vitro conditions. Throw as much light as you want at the Petri dish and optimize everything. How much can you get?
So the experiment that Shahaf showed you is an example where we had mitos and we had both of them working together at the exact same time. And in that scenario, we were able to decrease the activity of the ETC by about 50%. Now, we haven't gone through and optimized we can do that. We can do light doses, we can do expression levels, and probably make that even better. But as of right now, I think 50%. Now, the important thing too is that we have to remember that with that metabolic machinery that's there, that they're consuming, that they also play other roles. So when they're not being used and burned, they can go out and do other things within the cell, be used to generate biomass and for other processes.
And in how much do you think, how close to that 50% do you think you could get in C. elegans?
In C. elegans, I guess we really haven't thought too deeply about that. So our aging study, we noticed that the proton mode, the charge on your battery, if you will, goes down over time. And I'd say we are pretty close to that 50 mark in vivo with C. elegans as they're aging. But we can do more careful studies and actually quantify that.
Andrew, maybe you can also mention the experiment you have done with Roten on the other inhibitors of ETC.
That's another good example in our EMBO reports. We wanted to test to see what would happen under conditions where we would impair mitochondrial function, and we could give C. elegans rotenone, like lethal doses that would kill them, and we could turn their mitochondria on using our construct, and would allow them to live longer. So we could.
So Micah has a follow-up to this question.
Yes, go ahead.
Yeah, go.
Yeah, just wondering, have you guys tested or thought about testing these worms in a hypoxic environment? So they're not getting oxygen, therefore their normal ATP processes can't function properly, and then see how low you can bring that oxygen level down before the worms start dying?
I guess that's where my academic life lives, is in the neurons and hypoxia world. And we have those experiments ongoing right now, Because as you can imagine, with the system, we are no longer requiring carbon sources, and we're also no longer requiring oxygen. So aside from aging, there are many different aspects in which we could use this system to generate energy where we don't have as much metabolic input or oxygen around or conditions that are stressful. So those experiments are ongoing right now, and hopefully we'll have an answer pretty soon.
We also have another question from Cosmo. If you want to unmute.
Yeah, any idea what the quantum efficiency is with regards to like photons per protons?
Yep, so it's catalytic, and we need one photon to pump one proton. So it's a very quick cycle as well.
That's wild. Okay. And pretty efficient. You're not like generating lots of ROS.
So that's an excellent question. So with this system, we are not moving electrons. So the electron transport chain moves electrons down the chain, and we make a lot of ROS because it accidentally slips onto molecular oxygen. But however, in our system, we're just moving a photon. There's no electrons being moved by it.
Any follow-ups, Cosmo, or are you good?
Nope. It's good. It's good food for thought. I look forward to my future solar panel days.
That does suggest another follow-up, if it hasn't already been answered, which is what is the overall effect on ROS production for all these different techniques? Is it just like you said, that it has no effect? Or is there some way that if you put this light harvesting stuff in there and it's pumping these protons, isn't that going to ramp up mitochondrial, whatever you metabolism?
Oh, yeah. So I guess, yep. So that's where my other love lies, aside from bioenergetics. It's ROS production. And so we got to think of it in the context of we are decreasing the ETC. I guess the short answer is there's a lot of different contexts that we have to look at what happens to ROS production. And we have to look into that. We can speculate that by using mito-1, we're decreasing the activity of the ETC and thereby decreasing the electron flux and the possibility for ROS production. Now, ROS has this biphasic role inside the cell, where there's a little bit of ROS as a very hormetic response, and which can help us live longer. But once you cross some sort of threshold, it becomes more damaging, and that's where you start to see the shortening of lifespan. So our hypothesis is that mito-1 will decrease ROS, but again, each scenario is going to have a very specific amount of ROS, which could be linked to that phenotype. So some scenarios, ROS might be good. Other scenarios, a lot of ROS will be bad.
Okay, any other questions from anyone here for now? Otherwise, I'll ask our foresight round of questions. Just want to give it a minute. If you come up with another question, just feel free to raise your hand or drop it in the chat anytime. Otherwise, I'm going to ask you guys a few of the kind of questions that usually people find useful afterwards, but that are perhaps a little bit more general. One of them is, if everything goes well, where do you see this ending up in 5 years? Let's say everything goes just according to plan, but without any miracles or something, but you're just making progress. Where could you see this in 5 years?
So where do we see ourselves in 5 years? So
yeah, if everything goes well, like what's like the long-term potential, like the medium-term potential of this technology?
Yeah, so the way that we see this right now, we're targeting this to eyes and to light activatable, like tissue that light can reach. But as we've alluded to, there are many scenarios. There's heart disease, there's cancer, which can all be influenced by these changes in metabolism. And what we're doing here is changing the way that humans make energy. And there's tons of different applications for this. But again, we got to stay focused. So we're going to focus on this diabetic retinopathy, which is a tissue which is light accessible. Mitochondrial dysfunction is involved in that. And we're going to get some preliminary data with our CROs in order to help push that forward. And then from there, we'll be expanding and doing clinical trials in the next 5 years to see if this is applicable. But again, the application of this technology is very broad, and there are other approaches that we want to take in different sections and completely different fields, which can decrease the reliance on traditional oxbots. We're again like converting humans to solar panels, and there's a lot of things that we can do with it.
Yeah, there's tons of banter around this in the chat already. Okay, Aram, you're up next.
Yeah. Yes. I was wondering what kind of mutation can happen to that protein that gene construct that could actually pose a real danger in terms of safety to the individual. There could be somatic mutations, of course. There could be germline mutations and all kind of that. I think we have to figure out also not just whether the system is suitable, but also what could happen wrong in terms of mutations that can trigger something that is so central to the survival of a tissue.
Yeah, so that's an excellent question. So what could go wrong with mitoan if we expressed it, if it accumulated a mutation while it was in humans, of what would happen? So the biggest thing would be, I'd say that if we had a mutation that was bad, it would lead to inactivation of its catalytic activity. But in theory, we still have it. Remember, this is acting as like, I would say like your car that has both a battery and a gas engine. And if the battery went dead, you still have the gas engine that's there. And this is acting as a supplement. The worst case scenario that I can think of this is if we had some sort of mutation which caused an unfolded protein, which then led to stress, as something similar to any sort of proteostasis malfunction. But that's, I feel like, more of a multiple mutations that would happen.
Yeah, I'm afraid more, frankly, from the few moments that we got here, I'm more afraid of a disorienting mutation. That could be, I think, fatal to a lot of tissue. But it's, of course, all still academic. The whole thought is really interesting. I think you have a lot of courage, so I commend that.
And so the biggest one, the targeting sequence and the structural domains, is a big chunk of the protein. So you'd have to lose, if you lose that beginning part, that brings it to the, I guess the way to put it is the structural, your concern is if it's flipped the other way, it will cause uncoupling and energetic collapse.
Yes.
But in order for that to happen, you lose the very beginning of the protein. Mm-hmm. If you lose that very beginning of the protein, it's not even going to make it to the mito, because that's the part that drags it from the cytoplasm and gets it imported into the mito matrix. So in that sense, it could be viewed as a safety valve or a safety concern. So if that gets deleted, it's never going to make it to the mito to cause the mito damage.
You can still imagine recombination events that could still trigger that possibility, but making it as unlikely as possible is probably what you can do. Yeah.
Yes, agreed.
Yeah, thank you very much. Yeah.
Well, we had a kind of little bit of a thread going here in the chat on like dosing, and just like how much energy, I guess, can be generated like this. So I don't know if you want to take this up here. Seems like
so I guess I can comment on. I'm trying to see if I can find the actual comment, but so with light dose in our EMBO paper, we have the light dose response. So we changed the amount of light dose that it got and looked at the activity of it. And again, this requires one photon to move a proton. And in the mitochondria, that membrane, you actually don't need to move that many in order to generate that membrane potential. So you don't need to move that many protons in order to generate that battery or charge it. So we have those numbers and they're posted in that EMBO paper in one of the supplements. And one of the cool things that I saw on here too that we didn't talk about, which I saw, like, we can apply this to mitos where they are for mitochondrial disease is another aspect that we're looking at, in which there's a lot of mito diseases where the mitochondrial DNA is mutated and the electron transport chain doesn't function. Again, this is a construct that could help in those scenarios where we've already tested that, where we would poison the ETC and re-express this to be able to help.
So can we go back to the part of the talk where you expressed skepticism about tuning to wavelengths and thought that this light generation inside the body with a drug was a better way to go? I guess my strong intuition is the exact opposite, which Cosmo alluded to in the chat, right? Near IR can get, in lean people at least, can get to a large fraction of the body, whereas the idea of a few grams of drug generating large quantities of light just seems like it's not going to be able to come anywhere near the total energy delivery. Elaborate on why you were biased the other direction.
So there's advantages to both. So one of the things, the further you go, the more heat generation you get. And that's also with that infrared studies. Light is not necessarily benign. It can have effects as well. So we try to stay away too far red shifted because of the heat. The same way you don't want to go to the shorter wavelengths. So shorter wavelengths like UV, we all know they cause damage associated with aging. The same thing with blue light. Blue light will cause reactive oxygen species generation through endogenous photosensitizers. So there's a sweet spot where we need to be. And that wavelength of light will go about 3 millimeters through tissue, through complex tissue. So there are other ways to get around it. Maybe we can be red shifted and then also take advantage of some of these other systems where we generate the light right where we need it. You know, if we are looking for more blue light, maybe if we made blue light right at the source, or if we needed infrared light, maybe we can make infrared light right by that source.
Why is the heat generation bad?
So the heat generation is coming from the wavelength of light. So if you ever have your hand and put it in front of an infrared light or an infrared heating light, you can feel that heat being generated.
Sure, but why is that a problem? Like, humans do a pretty good job of
Thermoregulation.
Yeah.
Yeah. Again, some systems are just optimized at a particular. It could be a problem, and it's just an artifact that we don't want to get into too much, is what I would say.
Yeah, I guess where I'm coming from is just that humans evolved standing outside in the sun for 12 hours a day, and we get lots of infrared, near infrared, and we seem to do fine. And so it seems like we can capture that rather than just literally just using it for heat.
If
By
anything, there's
default,
more
heat
science
by
coming
default.
out showing that's actually healthy, especially for mitochondria.
Yeah, doesn't light already do all kinds of things to mitochondria?
So right now we have very indirect and correlative data showing that there's better activity of mitochondrial function. But again, we don't know the mechanism behind any of that. It could be a simple thing of heating up your sample and now your enzyme system is working faster. So if we put you at 15 degrees, you're going to move slower. We put you at 39, you're going to move faster. So the other
We have
comment.
lots of discussions in the chat and then
Yeah.
a new question, but I don't know, Shahaf, if you want to pick up the bit about us generating too much light. If not, we can have maybe Lynn ask her question. I don't know if you want this to be discussed.
In general, I'm very much available in LinkedIn. So if anyone has a question, you can contact me on LinkedIn. I think maybe I just want to allude to Caro's question, why I mentioned in the presentation that I would prefer the drug over the red light. It just, I think, in a simple answer, we have a clear idea to do it with the drug. And I think it might be a bit more difficult with the red. However, I've proven wrong a million times when I had any hypothesis.
So I guess what back in the envelope calculations about total amount of energy and translating that into what my earlier question was in vitro about percentage of the normal ATP synthesis that you can replace, which you said optimally up to 50%, right? With a drug, how much drug are we talking about taking? Let's say you're taking 5 grams of the drug per day. At most, what fraction of ATP could you replace? It seems like that's going to be a very small percentage with drug only and no inside body light generation, unless you're going to near infrared wavelengths and getting it from externally.
Yeah. Again, this is I think it's a great point to discuss and also a great point of our company's future, how to deal with this. I think we have an idea about the drug system that generates, I think, even too much light. You can think about microscopy. And as Andrew mentioned, 1 photon is 1 proton, which is quite a bit. But I don't think we can get too much into discussion with before we have any data to support either way. I think it's a really good point. And I think what I take from this discussion is not to cancel trying the red light, because it seems like the overall discussion point, we should really also go in this direction. So I really appreciate it. But I think we need more data. This technology is very new, and there are a lot of things we can still need to clarify. It's a good point. We need to really make sure, I think we also talk about it in ARDD. We need to know how much miton generate how much energy and how much is it in percentage of what we generate via food, as you alluded. I think it's an excellent point.
Yeah, and just
Okay,
build
we
off
only
of
have
that, right?
definitely reserve 1 minute for my last. Lynn, if you want to ask yours, now is a great time.
I was just curious to know if the mito technology is focused on prevention of vision loss or in combination with cell therapies to restore people's vision, or both, or just about a little bit about the delivery. Is it going to be an injection in the eye, or is it going to be with in conjunction with cells that are genetically manipulated?
So excellent question on, they're not mutually exclusive. So that's something that we're looking at in our C. elegans studies about how this works with other signaling pathways as well as other approaches. So we don't think they're mutually exclusive. We can do both at the same time. Now, the other reason why we chose to look at the eye is because the FDA approved AAV delivery of a transgene to the eye. So we're going to take advantage of that pathway that's there. So we're going to be using AAVs to express mitoan within the mitochondria in the eye.
Okay, wonderful. Maybe I'll even get 2 minutes for my questions. Cool. Unless someone else wants to chime in with something very last minute here. Okay, my question is always the same at the end of this call. It's, if people are excited about your work, what are your next plans and steps, and how can people support you best? What's missing? Like, what are challenges that you're facing? How do you wish to collaborate? And how can people contact you?
Yes, so we are working very hard that Andrew will finally have LinkedIn. And then I think both of us will be available in LinkedIn. And yeah, we are always open for discussions. Actually, the next 2 weeks are full of meetings on this. The best ways to support is to discuss with us, give us some ideas how to move forward. What are the disadvantages that you see? I think exactly with this discussion, at this point, we're also looking for funding. We have the seed funding to carry some of the experiments, but the more I think we get at this moment, we can have it quicker and faster, so we can have yes or no to go to clinical phase. So we will appreciate any support. Actually, I think our company is turning almost 1 month old, so it's actually very young. And yes, but I think you already supported us a lot by giving us the platform. And I think some people now are aware of this technology that they were not aware before. So we already appreciate the support.
Wonderful. Then I hope that people will follow up on you. It sounds like there's a ton of interest and appetite for further discussion. So yeah, please feel free to reach out to, I think you both dropped your contact info also here in the chat. Thanks a lot for joining. Thanks everyone for your really interesting on the record questions and just, hilarious chat discussion. I hope you have a good one, and I'll see you all very soon.